Welcome. My name is Steve McConaughey. I'm the Executive Director of the Global Nerve Foundation. Today, I have the opportunity to speak to Curt Deister, the Senior Manager of Neuroscience and Innovation from our founding partner, Axogen. Curt's going to speak to us today about the evolving technology within the nerve space, as well as his personal journey from chemical engineering to product development. Welcome, Curt. Thank you for being here today.
Thank you.
And I was wondering, maybe to get started, if you could maybe just give us a little bit of your background and studies and where you're from.
Sure. So I grew up in the New Orleans area and then went over to Texas for undergrad and grad. I went to Texas A&M for undergrad and then naturally drifted to UT Austin, no problems there, for graduate school.
Okay.
And then that was where I became involved with Axogen. So my last year in grad school was actually under most of it anyway, was under a sponsored research agreement. And that was process development for the advanced craft and getting us towards launch there. So we took two technologies, one that I had been involved with that had been going on in the lab I was in prior, a decellularization technology, and matching it with some technology out of the University of Florida, removal of some inhibitors. And we paired those together and made the advanced craft, and then came over to work for Axogen full-time after that, working on some process improvements and so on and so forth.
So a couple things there. So I feel like UT Austin seems to be a very popular place as far as kind of science leading to industry or kind of development or businesses coming out of some of the business there. So when you went there, did you go with the intention of kind of being able to work in that transitional space or translational space between science and industry, or did it just kind of happen when you were there that the project was available and it was something that you were interested in?
So I think, especially coming from a chemical engineering background, and A&M even more so than most other places, is very, this is the textbook you need to do the problem at the plant. It's very industry focused. So I had that background, but I didn't have it all planned out or anything like that going in. Nerve, as you know, has been kind of the redheaded stepchild of the medical world for the last really about 120 years. So I did not have that planned out, but it's in the background, and with Chemi and with Texas in general, UT Austin, I mean, for Chemi, good enough, so much battery, and he was there at the time I was, glucose sensor, other people. So there was a lot of commercialization, and I think that's a very good thing.
Right. Agreed. I feel like it's a way of thinking that a lot of people in science aren't necessarily exposed to, that commercialization idea and that idea of kind of taking it to the next level or getting it out of the lab.
Yeah. I think that is important. If you're not focused in on something that's going to help your end user, in a case a patient, you can get completely disjointed and you can get lost, because reality is infinite, and you can get lost. It's very easy to do. And that's not helpful. That's not, you never know if you win, right? You never know if you're doing what you should be doing.
Right. No, that's a great way to put it, and why people think there's sometimes a disconnect between academics and the real world. And I think that that connection, it really helps.
Yeah, you definitely need the basic science to get understanding and be able to get the tools to apply. But as I mentioned, the background, especially engineering, is, I think, much more focused in on solutions. That's kind of inherent in the name in the field.
So moving from chemical engineering into this type of project, was there a biological aspect that you kind of picked up during graduate school, or were you still kind of aligned more on the chemical engineering side?
So I knew I was going to be on the more bio side
Okay.
of chemical engineering, and wound up being on the very bio side of chemical engineering. Um, Maybe not the right choice of words there, but um, what the chemistry you're using, it's more knowing what to look out for,
Got it.
and knowing the background of things. And if we did X and Y, so that means we created A,
Right.
or we had a risk of doing that. Um, but you're not, I'm not dealing with the vapor pressure of something on a regular basis. I'm not trying to figure out where to put a pipe in a distillation column to get the right cut. Um, Other things like that.
Right.
Uh, and a lot of biology tends to be less quantitative. So just going through a couple of things that are different than in the reality of making a product, especially a tissue-based or biologic
Mm-hmm.
product, cell-based product. You don't have a nice cell healthometer. It just doesn't wind up working like that. You're looking at um, histology. You're looking at,
Right.
hopefully you're leading towards something that's very well grounded and very quantitative, but it's a long way to get there.
Yeah.
So, and then you have other weird things. Like I never thought I would be doing microsurgery, but rat sciatics are small and you can't do them, by naked eye. So you have to learn that to be able to debride nerve, to be able to
Mm-hmm.
suture it in place, etc.
No, that makes sense. So you used a few terms early on about the project and what you were working on. Maybe you could elaborate a little bit more on those, as far as I believe it was the decellularization,
Right. So,
and some of those methods.
yeah. So a prior student had started a project on a optimized decellularization process. So there's been a lot of tissue decellularization in the past, I don't know, since about 1950s. I
Okay.
guess it started and has been on from there. And there had been several papers about nerve grafts specifically. And you wanted, if you don't decellularize and you leave all of the cellular materials in there, you get immune rejection.
Okay.
So
Got it.
you have to extract that out. And then sometimes you can even get away with xenogenic material, where you've removed enough that the body isn't going to really get that angry and do something bad. Sometimes that works, sometimes it doesn't.
And that's if
So
you're crossing species. So
right,
if you're going, okay.
right. There have been a lot of that in headlines over the last couple of months,
Right. The
and
heart
it's
transplant.
yep. And it,
The
unfortunately, it would be great if something like that works. I'm sure it will eventually, but there are a lot of steps along the way
I see.
to dial all that in. But if you think from going back to standard engineering parlance, you have bottom up and top down design. We can take things out, and that's one way of looking at it, and then we can add things in.
Got
So
it.
that's top down, then bottom up,
Okay.
and a lot of ECMs tissues are actually fairly well suited for that. Because you have cells and you have extracellular matrix, and there's a big solubility difference between most of the things in there.
So that
So
makes it easier to remove
right,
different
so you
ones.
can
Okay.
try to target where you don't harm the things that are in the ECM, but you
Got
take
it.
out the cells. You take out the cells, you take out the most variable material, the things that are most likely to be antigens,
Okay.
and you leave what's the difference between your collagen and my collagen is pretty small. Even the difference between porcine collagen and my collagen is relatively small
Okay.
compared to the difference in, say, our HLA antigens.
Got it. So less chance of a reaction.
Right. And a milder immune response, because you always have an immune response, you always have inflammation, and you need it to drive remodeling and regeneration.
Right.
So that was the project, going through and optimizing a process to best preserve the ECM, and in nerve especially, you have a very fine detailed ECM endoneurial tubes are that, almost, micron or less than a micron thick for the basal lamina, actually. So leaving that intact, but taking out the cellular material.
Okay.
So then the rest of my PhD was more looking at growth factor combinations and interactions between ECM and growth factors, trying to see if there were optimums that we could use there, and then came back into the decellularization work with Axogen.
Got it. Got it. And we always say that, science is a very collaborative process. I'm guessing at UT, you had a lot of collaborations within your graduate work. And I guess I'm wondering, for people who might not have as much experience in industry or haven't been involved with an industry organization, what does collaboration look like there? So at Axogen, do you guys have a lot of external collaborations or even just internal collaborations within the teams that you have, as far as still kind of working and pushing forward in the R&D?
Yeah, the more things change, the more things stay the same, right? The first thing that you have to ask, but it's very helpful when you do. So there's some different dimensions when you're dealing with academic collaborations. It's somebody who knows a particular, say knows a particular chemistry well, knows how to make
Right.
a polymer that you want to make into a scaffold, or knows how to isolate one compound, or has already isolated and you just want to use it. They don't know how to do animal work, you do.
Yeah.
Or something like that. You have some
Right.
extra dimensions when you start getting into commercialization, you can start getting into industry. The first one that it's a big deal in academia as well, but it, in my opinion, can also often get overlooked, is you have to stay with the surgeon. You have to stay with the problem. The surgeon in our case is somewhat a stand-in for the patient. You have to stay with the problem and know what it is, because it's usually not very well defined. That's why somebody doesn't have a solution. So you don't know what it is. You have to keep looking, and you have to keep a touch point there. But you've got other things like regulation, manufacturing. You need to pay more attention. The more you've put in, the more attention you need to pay to intellectual property.
Yeah.
And you don't want to get right to the end and find out that your collaborator is commercializing the same thing as well.
Right. That's a great point.
And that's in academia, you can both be working on something in parallel and everything's fine. That's what everyone's doing. But at some point, the more other people's money you're using, you have to pay more attention to that. There is a fiduciary responsibility with that.
That's a great point. I feel like with academia, it's very much the end goal is really kind of the publication, right? The publication, and then maybe the next grant. So I feel like
or maybe vice versa. But
yeah, right. Great point. So I feel like the timelines are much shorter, right? Because you're just trying to get to that point, and then you move forward with the next project and the next collaborations. Whereas, with industry, it could be much longer, as far as from the start point to the end point. If you figure the end point is kind of getting to market, you're dealing with much longer timelines and dealing with a lot more, like, as you said, complications.
Yeah. There are more things there are just more things to be aware of there.
Right.
But you still have in the industry, you still have to do some of the same things as far as finding either a manufacturer who can make that particular polymer, or go talking to the researcher who understands that particular chemistry or that particular other thing that will allow you to do what you're trying to do.
No, that makes a lot of sense. So it is a little similar in finding that it's kind of skill driven, as far as what you're looking for, who you might reach out to, things along those lines. What about collaboration, like within Axogen? Like, what does it look like for research and development within a research project of Axogen? Because I know you all put so much resources and time into developing products to improve care. So what would it look like as you're kind of starting with an idea and working through that process?
Yeah, so every project is different or is imperfect in its own way. But you need to identify the problem,
Yeah.
and that needs to stay up front and center. And then you figure out what part of it you can um, make less of a problem, right?
Mm-hmm.
What are you going to do? How are you going to do it? And then can you make it? So in that, there's always a back and forth between, can I do it? Is it worth doing? But then in some ways there's another point there is it profitable?
Right.
Because worth doing, there are a lot of things that are worth doing, but you'll never get paid for them. You can really only do that once.
Right.
And then you're out of money. So how do you kind of bounce around in there? It's a cycle. What we do is it's termed a stage gate system. So you do the work up to one point, then you review it. And do we have
That
a
makes
market?
sense.
Do we know what it is?
Yeah.
Would we have to do something that we can't do or is too expensive? We don't have it in the budget for the next 3 years, so we probably shouldn't spend this money now. Or, for instance, do we have something we would need an entirely new sales force to engage to be able to sell it? Well, that's really expensive.
Right.
So maybe you don't go that way. Maybe something's more valuable that utilizes your existing sales force, or etc. Because there
Yeah.
are
Yeah.
a
Yeah.
lot of different things in there. So that's a kind of a very wordy answer to say we go in stages. First thing is get the problem defined. You want to take out the big risks first,
Mm-hmm.
and then you get into the details. So the first part is fail fast. And then the second part, after you get through a couple of stage gates, it's make sure you don't fail. So there's a big mindset change between the beginning
Right.
and the end. By the time you're going into a sterilization validation, you should have that locked down. You should not fail that.
You have to get all that failing out of your system early on, and
right.
then once you're through that, you're good to
right.
go.
You don't want to fail right after you invest. You invested the most money and then you fail.
Yeah. Yeah.
So you want to fail early, and you can fail often as long as it's early.
Right. No, that makes a lot of sense. And I think that's I think that's true even in a lot of research. I guess it's a little bit different though, if you're dealing with those large grants and you're trying to just get to the end of it versus in that regards, as you said, you have a lot of other restrictions and forces that you kind of have to worry about and balance. So yeah, it's interesting. I like that fail fast. That's a really good way to put it.
I think in both academia and industry, you need to learn from your mistakes.
Right.
But the trick, I think a trick in academia, not that I am one, but is being able to publish your failures.
That's true.
Whereas,
still getting something out of it and being
right.
able to write. It's almost still having some success out of a failure by having a publication or something you can show for it.
One publication is one publication. So if
right.
if you get the same amount, you get the same amount. And you set up the next grant application.
Right.
It's at some point.
It's a good point.
Not having a successful product is just a zero.
Yeah. No, good point. So we kind of touched on it, but we didn't quite get to the end of the story. So what you mentioned kind of coming to Axogen to continue to work on the product that you were originally working on at UT. Can you maybe just give us a little bit more background on that and kind of where that particular product ended up?
Sure. So that was the advanced nerve graft, or is the advanced nerve graft. So came over with the sponsored research degree in Macarius to the first commercial process. Then we did a major process improvement series in the first year of commercialization. And we've been pretty much with that since. We've added a number of back ends, bells and whistles, are with the change from regulation as a tissue to a change in regulation as a biologic. That is one of those big risks I talked about before that we didn't find out about until much later. And it hurt. But those are, I mean, that's reality. You have
Right.
surprises. You don't know
Yep.
what you're getting into until you get into it. But so yeah, that was that product.
Okay.
Came
And
over.
what does that for the people who might not be familiar, what is the advance, and what, like, maybe just kind of tie it back to the what you were working on as far as the decellularization and other techniques that you were kind of working on at UT.
Right. So the advanced nerve graft is a human allograft nerve, decellularized human allograft nerve. That's commercial off the shelf. So it can be implanted instead of harvesting an autograft, which to maybe annoy some people, but I like to call that more of a nerve injury transfer, because you're definitely making a nerve injury and you're hoping to get recovery in a different location.
Got it.
So we have an off-the-shelf graft that you can do a repair without doing any other damage.
Okay.
The core of that is taking out the cellular material and leaving the ECM behind, and an active ECM at that. So the laminin is active. And also removing an inhibitor. Chondroitin sulfate side chains actually block the activity of laminin as it interacts with cells. So that's tied in through the scar in the CNS, but it's also present in the PNS. Kind of like guardrails along a road. It keeps the axon where it's supposed to be. But with grafting, especially, it helps to remove that so things can find their way.
That makes sense.
So we had to scale from rat grafts, which had all been the preclinical work had been on, which is about 10. You're pushing it at 15. You can get to 20, 25, but it's hard. You're going through a lot of rats, and 1 millimeter by, let's call it 15 millimeters. And we're scaling and we're launching 4 to 5 millimeter in diameter, and eventually 70 millimeters, a little over 70, but the tolerance long. It's about, can't remember the math right now, but it's over 100-fold larger.
Right.
So you're dealing with multiple orders of magnitude difference just in size. That has a lot of differences in what you have to do. And also, you just think about the histology. The histology is now different because you're dealing with a whole different size. It has to be in a different processor, et cetera, to get quality grafts. Sorry, histology sections. You're not rat tissue, so the antigens are different. You have to work up different antibodies. And all of that, we did most of it early on, and then we just continued to do it as we went. I scaled up to get better yields and less effort.
Got it. So the processing side of things.
Right. And then you learn things along the way about how to do things or what to avoid, things that you don't see in academia, like particulate levels or bioburden levels almost never come up in academia. They come up all the time in real world manufacturing. You have to pay attention to that stuff, and you have to go back through each piece. You're using some stainless steel tool that rusts, that's creating particulate, that's messing things up.
Wow. Yeah.
Things like that. That was the totality of those several years worth of initial and then subsequent work.
Got it. So you kind of moved into nerve space you didn't really go into UT thinking of peripheral nerve, right? And started working in the nerve space. So starting at that point, what kind of surprises you the most as far as where it's gone in the time that you've been working in this field? Is there anything that you just kind of didn't see coming, or were surprised when that when that kind of came along, or maybe it's a particular question that you're trying to answer?
So the thing that surprises me most about nerve is kind of in a negative light. You can go pick up Sunderland's book that's 100 years old, um, and a lot of the things he's talking about are still under active research today.
Right.
We've made a lot of progress in some ways, and in other ways, we've made very little progress. And that's kind of some of the logistics of that, are in a way some of the most exciting things to me about nerve care, that I think we're building up enough mass that it may be able to be treated as its own specialty, or at least subspecialty, as opposed to just kind of people find their way,
Right.
and that will give a better quality of care. And just like we've got all the other specialties along the way, and it's not just medicine,
Yeah.
which then you get your silos and it has its own problems. But nerve is a different tissue. It's the only tissue in the body where the cell that you're trying to get to repair itself is a meter away in the hand, from where the injury is. No other tissue in the body is like that.
Yeah, that's a good point.
And so it has differences, and if you respect them, you get better results, or the patient does.
Point. Right. So then looking ahead, where do you think the most potential, or what are you most excited about as far as in this space, besides is there a particular question, or is it really just getting kind of into its own field and nerve kind of standing on its own?
So I guess I'm more excited about what I don't know, and what hopefully, dealing with industry, it's usually something that's simple and relatively well known. Remembering attending a talk, forgetting the guy's name now, but he invented PCR, and everyone's like, "Oh yeah, of course that will work," after he did it, right? So things that are known and you can apply, and it doesn't have to change the world. I'm thinking cup holders in cars. It actually improved the user experience substantially. And it's just a little piece of plastic molding. So when we can do things like that, and be able to interact to do that, so know the field and be able to put something in there that works. That's where I think we can get some real good things happening. Plates and screws were not new at all. Even the technology to do most of the base technology, if you will. I know there's a lot of everything, but in joint implants, I mean, stainless steel has been around for a long time,
right?
But it's getting it together into the right useful thing. And I think
that makes sense.
All that pent up research over the course of 100 years. There are things we can do, and excited to be able to try to commercialize those and get them to patients.
Yeah, absolutely. I agree. I think the potential is huge, not only within the nerve space, but then bringing in those other technologies that are kind of being developed in parallel. So it's going to be exciting to see where it goes. Then I'd like to thank you, Curt, for the time today and kind of walking us through what it looked like getting something kind of from the lab all the way through on the industry side of things, and kind of the comparing the back and forth of academia. It was really helpful and very enlightening. So thank you so much for your time.
Yeah, thank you for having me.